the proceeding talk is from Lisa Guy from UCLA. She's going to present a paper about finding associated variants in genome-wide association related to multiple traits.
So I'd like to thank the organizers for giving me this chance to present our work. So this is a project for detecting associations using genome-wide association studies on multiple traits. I'd like to start out about kind of motivating why we're looking at multiple traits. So the vast majority of GWAS have been conducted in a single trait at a time. A lot of you might recognize this image. It's kind of a perennial favorite in a lot of different GWAS talks. It's just a GWAS catalog, and each different dot is a different SNP which has been associated with some trait in GWAS. And the reason why you might be interested in multiple traits is a phenomenon known as pleiotropy. So pleiotropy is where a gene may affect more than one, where a gene affects more than one trait. A lot of recent work has shown that pleiotropy is actually extremely widespread in the human genome and includes traits which may or may not be so obviously genetically related. So for example, it's been shown that there's pleiotropy between multiple sclerosis and schizophrenia, but not bipolar disorder. And kind of the intuition here is that analyzing traits which share genetic basis together may increase power to detect variants associated with such traits. But the thing is, what's the challenge here? So, as I mentioned before, it's not always obvious which traits actually share a genetic basis, and the degree to which they share a genetic basis also varies. So, for example, in these Mahan plots for 3 different traits, cholesterol medication, and use of insulin medication, you can see that there are some SNPs which are significant in all 3 traits, but there are others which are only have some signal in 2 out of the 3 traits, or even 1 out of the 3 traits. So just kind of throwing traits together may actually cause you to lose power, because not all variants will be associated with all these traits. And this is what a traditional meta-analysis method would do, right? It would have basically assumed that there's an effect in all studies in your meta-analysis. So our goal here is to instead develop kind of an analysis method that allows for some flexibility. We want to be able to detect associations even when the variant is only associated with maybe a subset of traits in the analysis. So first, some background about how a genome-wide association test actually works in a single trait. So what you want to do is you want to estimate the effect size of a variant on some phenotype. So let's say that we've collected a bunch of phenotypes in different individuals. That's the Y1 through YN. And we have also collected genotype data. Those are the X1 through XN. And we want to estimate the effect size beta, assuming a linear model. And we also have some error term that's non-genetic. So for example, if we were doing a GWAS and HDL, we would have HDL measurements, and we would want to find what's the effect that our SNP of interest has on HDL. So the summary, another thing that I'd just like to mention is that our method uses a z-score, which is just a scaled version of the estimated effect size. I'm not going to go into the details here. It's just normal linear regression stuff. You'll have some estimate, which is the beta hat here. It'll have some variance. You can just rescale it so that you get a z-score, which has variance 1, and it is a standardized estimate of the effect size. And we just do that because the studies in your analysis might be different sizes, and doing this kind of scaling just helps make them more comparable. So what if you wanted to do association with multiple traits? So how does that work? So kind of the standard thing that you would do is just to basically look at each trait independently. So just basically looking at one trait at a time. So for example, maybe you have GWAS summary statistics, so that's our z-scores from a SNP in 3 different traits, HDL, cholesterol, LDL, cholesterol, and triglycerides, let's say. So the standard analysis where you look at each trait separately, if you wanted to answer the question, is this SNP associated with at least one of these 3 traits, you would just look at the maximum GWAS, or the most significant z-score out of these 3 traits. So in this case, it would be the LDL z-score. And if that one crosses the threshold, you know that SNP's associated with at least one trait, and then you just kind of ignore the rest. So we're going to call this standard analysis multiple independent GWAS, or MI-GWAS. And another thing that I'd just like to mention now is that when you do this kind of association testing in multiple traits, you have to do a multiple testing correction in order to control your false positive rate, because you're basically giving every SNP multiple chances to meet a significance threshold without this correction. Okay, so then let's move on to our method. So in our method, we want to allow for, remember, we want to allow for heterogeneity across traits. So what we're going to do is that we're going to use something that we're just calling a configuration vector, an activity vector, where we, in our model, we assume that a variant either has an effect or not, that is, either is active in a trait or not. And we can model this using this configuration vector, which is just a vector of zeros and ones, where it's a 1 if it's active in that trait and a 0 if it's not. So just for example, let's say that we have a SNP which has an effect in HDL and LDL cholesterol but not triglycerides. If using the z-scores we saw from the last slide, it wouldn't be that surprising if this was a SNP which was active in HDL cholesterol and LDL cholesterol but had no effect in triglycerides, like no true effect in triglycerides. Our method is called CONFIT, because it's configuration-based testing. So the hypothesis that we're testing here is whether the SNP has an effect in at least one trait. So our null hypothesis is that the true configuration is the null configuration, that is a configuration of all zeros, like no effect in any trait. And our alternate hypothesis is simply that the true configuration is not the null configuration, so there's at least one in the configuration. So what our test statistic looks like is that it's a weighted likelihood ratio of all the different possible non-null models against the null model. So in the numerator, we're just summing over the set of all non-null configurations, and the weight is a prior that we put on each configuration. So talk a little bit more about the prior. You might have kind of, you might expect some configurations to be more likely or less likely in your data than others. So you can use that to weight each model in the test statistic. So for example, you could use functional information here or what have you. So we set the prior based on the GWAS data. So what we do is that we count SNPs with a GWAS p-value less than some threshold in the same subset of traits that are active in that particular configuration. So just as an example, let's say we have 3 different traits and we have a few SNPs from each one. That dashed line shows our threshold for the prior. And, oh, it's kind of faint, but you can see that there's 1 out of these 10 SNPs crosses the threshold in all 3 traits, so that would correspond to the configuration 111. There's a SNP that crosses the threshold in 2 out of the 3 traits corresponding to the threshold 110, and then there are a few that correspond to the configuration with only a single active trait. Okay, so we evaluated our method in simulated and real data, and I'm going to talk first about how we did the simulations. So what we do in the simulations is that we first draw the true configuration for each simulated SNP, and then we draw the GWAS z-scores from a normal distribution. So for example, if our true simulated configuration was 110, we would draw z-scores where the mean is 0 in the 3rd trait, which has no true effect, but we have some non-zero mean for the first 2 traits. So we find that compared to MI-GWAS, our method increases power when the SNP is active in multiple traits, including the scenario where the SNP is only active in a subset out of the traits. So for example, when we did simulations in 3 traits, and even when the effect is only present in 2 out of 3 traits, CONFIT still increases power. And another thing to note is that CONFIT still managed to recover the majority of SNPs found by MI-GWAS. So the power is comparable when there's only 1 true active trait, but we increase power when there's activity of multiple traits. So just to kind of get some intuition about how CONFIT's working, these are just plots showing the rejection regions for CONFIT. So each point is a different non-null SNP in the simulation. And what we have here are the black and gray SNPs were either found significant by both CONFIT and MI-GWAS, and the gray SNPs were found significant by neither. And then the blue SNPs are the ones found significant by MI-GWAS, which were missed by CONFIT, and the red SNPs were the ones found significant only by CONFIT. So the blue ones might be hard to see just because there's not very many of them. And then you can kind of see that a lot of these SNPs found only by CONFIT are in the corners, where each SNP, there is some GWAS signal at each SNP, but each trait by itself is not significant enough to meet the GWAS threshold. So this kind of matches our intuition for how CONFIT should be working. If we see signal in multiple SNPs, we can kind of combine that information and find the association. So next we applied our method to the North Finland birth cohort, which is a very well-studied classic GWAS data set, which includes multiple metabolic traits, about 300,000 SNPs and 5,000 individuals. We compared our method against a previous GWAS conducted by Kang et al. In 2010, which looked at a single trait at a time and just does standard GWAS using a linear mix model. And we also compared it to a alternate multi-trait method called MVLMM, which uses multivariate regression. So that is, it works on individual level data instead of summary statistics when doing the joint analysis. So we compared it to the same 4 traits used by Ferlat et al. In their multivariate regression analysis. So that is C-reactive protein, HDL and LDL cholesterol, and triglycerides. And one thing that I want to note is that in the joint analysis by Ferlat et al., they only found 1 significant loci using these 4 traits. So we found that CONFIT outperforms MI-GWAS and MVLMM on the Northern Finland birth cohort dataset. So we find all loci reported by Kang et al. In the original univariate GWAS analysis, including a loci which would not have been considered significant if they had done multiple testing correction for testing multiple traits. And we also find an additional loci on chromosome 8, which was not associated with any of these traits in the original study, but was actually replicated in a larger study and found to be associated with triglycerides, which is kind of a nice validation of our method. Next we applied our CONFIT to a more novel data set. So this is the UK Biobank. It's a much larger data set containing about 10 million SNPs and 300,000 individuals in a wide variety of traits. And there are also publicly available summary statistics for the UK Biobank provided online by the NEAL lab. So we selected just 4 metabolic traits kind of related to the ones used in our Northern Finland experiment. So we used self-reported high cholesterol, use of cholesterol medication, use of insulin medication, and elevated blood glucose. So we found that CONFIT still finds the majority of SNPs found significant by MI-GWAS, and additionally we find 6 novel loci. And one thing that I'd like to point out about these novel loci shown here is that they all have GWAS signal in both the high cholesterol and cholesterol medication trait, but they are not anywhere close to significance for insulin medication and glucose medication. And again, that kind of matches our intuition about which kinds of SNPs we would expect CONFIT to be able to find. So in summary, I've presented a method, CONFIT, which leverages the widespread pleiotropy across the genome while allowing for heterogeneity across traits in order to increase power to detect associations. We validated our method on simulated data in the Northern Finland birth cohort dataset, and we are able to find novel associations in the UK Biobank in a variety of metabolic traits. We've received many potential applications of CONFIT due to the widespread availability of GWAS summary statistics in a wide range of traits. And if any of you are interested, our code is available on GitHub for your perusal. So I'd just like to finish off by thanking all of you for your attention, and also I'd like to thank our lab at UCLA, especially my advisor, Eleazar Eskin. This was our joint work. Thank you.
We have time for one question.
Can you give us a quick sense of how this would compare to some of the other multi-trait approaches? Maybe MTAG, multi-trait analysis of GWAS, adaptive Fisher, something like that?
Okay, so there are a few other multi-trait methods that I guess I'll start off with some older ones. So one that's very common is multivariate regression, which I mentioned, and traditional fixed effects and random effects analysis. So those methods are different in the sense that they assume that the effect is present in all traits. And additionally, multivariate regression requires individual level data. So you mentioned MTAG in particular. MTAG definitely seems like a very powerful tool for multi-trait analysis. It came out after, I think it came out after we did. But the difference there is that I think MTAG, the focus is more on accurate estimations of the marginal SNP effects rather than increasing power. And another thing is that MTAG also makes the assumption that there is an effect in all traits, as opposed to our method where we kind of assume SNPs are kind of drawn from this mixture model, where in some traits they come from the regular like Fisher polygenic whatever model, but in other traits they just simply have no effect, whereas MTAG assumes that there is some effect in all traits.
Okay, let's thank again the speaker and move on.